Okay, in this lesson we're looking at the absorption of the products of digestion. In 6.9 we looked at enzymes and digestion with the hydrolysis of macromolecules into micromolecules. So by the end of this lesson we want to be able to describe the structure of a villus, and we want to be able to talk about how the amino acids and monosaccharides are absorbed, and also how triglycerides are absorbed. Here we have a light micrograph of a section through some villi, showing their large surface area there. And then if we relate that micrograph to a diagram and look at the cross-section of that villus, we can see the cells that make up the epithelial lining of the villus, the rich blood supply there, and of course in the middle of that, a lacteal that goes in and comes out of each villus. Now later on we'll be talking about how cells deal with the hydrolysis of triglycerides, and we're talking about these cells here on the outside. These are the cells that contain the endoplasmic reticulum and the golgi apparatus. These are the cells that we're referring to. Here's another diagrammatic representation. Again, here's the cells on the outside and the large surface area. We can see that the villus is made up of all these cells. These are the epithelial cells. These are the cells that have the endoplasmic reticulum in, and we've got our rich blood supply and our lymphatic vessels. And here we go. Each one of those is one of those, and the microvilli are those little extensions of cytoplasm on each cell. They actually are the microvilli on the villi. And here's another diagrammatic representation of a villus, and it takes into account the oxygenated and the deoxygenated blood, because obviously this is where we're having absorption all the way around, but because we've got a lot of active transport going on, oxygen will be being used up, and so will glucose. Here's a model of a villus. Here's the epithelial cells all the way around, and of course they are served by this blood capillary network. And if we took a slice of that, that is how it would look. And in the centre there, you can see the lacteal. So the villi are the interface between the outside of the body, meaning the lumen or our cavity, and the inside of our body, the blood and other tissues. And it's a highly specialised exchange surface for the absorption of the products of digestion. So let's have a little think. Could we complete this table where we relate the structure of the villus to its function? Before we move on, stop the video, copy the table, and see if you can complete it. Well, the first thing that we would say is that the villi are numerous, which creates a massive surface area. Secondly, they are thin-walled. In other words, the epithelial layer creates the idea of a wall, and that is very thin to reduce the diffusion distance. The next thing is that they contain muscle, and so they're able to move. This helps them maintain a diffusion gradient because the muscle mixes the contents of the ileum, and that ensures that as the products of digestion are formed, they're absorbed and moved along. And as it's moved along, new products from digestion are available for absorption. So here's an example of what you may have written. In our previous lessons, we looked at the absorption of glucose and amino acids by the ileum, and we talked about how they're absorbed by diffusion and co-transport. And now we're looking at the absorption of the products of digestion of triglycerides, i.e. namely monoglycerides and fatty acids. And we need to remember that lipids have been broken up into smaller droplets by the process of emulsification in order to increase the surface area for the action of enzymes, namely lipase. Now, once these products of digestion have been formed, whether you're going to call them monoglycerides and fatty acids or glycerol and 3 fatty acids, either or, both of them are fatty in nature. They are both hydrophobic. And the lumen of the gut is an aqueous environment. It's a watery environment. Now we want to keep these products of digestion separate because they're small enough to be able to pass straight through the epithelial cell membrane. However, the lipid droplet is in itself hydrophobic. And so what happens is the bile salts have both a hydrophobic part and a hydrophilic part. The hydrophobic part stays in contact with the lipid, and the hydrophilic with the watery aqueous medium of the lumen. And this stops the lipid particles forming together in a big lump, because we want to keep them small enough that when they reach the epithelial cell, they're able to diffuse through the plasma membrane. After all, this is made from phospholipids. So the bile salt stays associated with the lipid droplets, and they're now called micelles. When they get to the epithelial wall, the bile salts drop off, and the lipid, the fatty acids and the monoglycerides is able to diffuse through. And once inside the epithelial cell, we want the monoglyceride and the fatty acids to be recombined to form a triglyceride. So that takes place in the endoplasmic reticulum. So going into the endoplasmic reticulum, we have monoglycerides and fatty acids, and coming out the other end, we have triglycerides. They are then passed to the Golgi apparatus. In the Golgi apparatus, the triglyceride is modified. Cholesterol is added, and also some lipoproteins, and that structure is then called a chylomicron. The chylomicron is a special particle adapted for the transport of lipids. It's got a phospholipid layer on its outside, courtesy of the Golgi apparatus, and inside are lots and lots of triglycerides, of cholesterol with the odd protein. Once the glycerol and fatty acids are recombined to form a triglyceride, they're passed to the Golgi apparatus, they're modified with cholesterol and lipoproteins, and then they're packaged. Then they're ready to leave the cell by exocytosis and enter the lymph vessel. What we mean by this is they enter the lacteal of each villus. They don't enter the capillary network of each villus because, of course, blood is an aqueous solution. Later on, where the lymph meets the blood vessels and empties its contents into it, the chylomicron will indeed enter the blood. It will circulate around in the circulatory system until it gets to a cell that contains an enzyme on its outside which will digest the chylomicron further, and the products of digestion will be absorbed into the cell. Stop the video and use this list and see if you can confidently describe the pathway taken during the absorption of monoglycerides and fatty acids. For me, I would talk about the micelle being a particle of monoglyceride and fatty acids that are still associated with their bile salts. That on reaching the epithelial cell, the bile salts become dissociated, and the monoglyceride and the fatty acids are then absorbed into the epithelial cell by diffusion. On reaching the endoplasmic reticulum, they are recombined to form triglycerides, which are passed to the Golgi apparatus. In the Golgi apparatus, the triglycerides join with cholesterol and lipoproteins and become packaged to form a particle called the chylomicron that then enters the lacteal of the villus. That then flows into the lymph vessels and later on into the bloodstream. Once in the blood capillary, it reaches an endothelial cell. Another enzyme hydrolyzes the triglyceride. The products break down and are reabsorbed into that endothelial cell. Okay, so let's have a look at some exam-style questions. First one is straightforward. It just asks you to label the processes A and B. We're talking, of course, here to the diagram that I drew on the paper earlier. The second one wants the advantages of both lipid droplet formation and of micelle formation. And the third one wants you to name structure X and describe how it's involved in lipid absorption. Now, question 1 is straightforward, but question 2, it asks you for advantages of lipid droplets and micelle formation. So you need to refer to both of them, especially as it's worth 3 marks. The third question asks you about structure X and how it's involved in the absorption of lipids. And we need to make the point here that by the time the monoglycerides and the fatty acids have reached the Golgi apparatus, they've already been through the endoplasmic reticulum and they've also already been recombined. So we need to ensure that we talk about triglycerides entering the Golgi apparatus and the chylomicron exiting the Golgi apparatus. So let's recap on the main points. Well, we've got the first point that the emulsification of lipids into droplets increases their surface area for the hydrolysis by lipase. Also, that when fatty acids and monoglycerides are produced, they remain in contact with bile salts until they reach the epithelial cells of a villus. And that's because they are non-polar and the lumen of the gut is an aqueous environment. So until they reach that plasma membrane of the endothelial cell, we want them to not recombine as a big blob again, and we want them to stay separate. The fatty acids and the glycerol or the monoglycerides diffuse into the epithelial cell, and that's because it's made of the same substance as the plasma membrane. The fatty acids, glycerol and monoglycerides are built into triglycerides in the endoplasmic reticulum. From there, they go to the Golgi apparatus, where they are combined with protein and packaged into vesicles for export from the cell. The export from the cell is by exocytosis. The particles are now called chylomicrons, and they enter the lacteal of the villus, and then they enter the lymphatic system. And later the chylomicrons will flow into the lymph vessels and then into blood vessels, where eventually they'll be in a capillary and they'll come across a membrane and they'll break down and then diffuse into a cell that the blood capillary serves.